Hello, everyone. Thank you so much for joining today's webinar titled Breaking the Cold Chain, Lyophilization of Lipid Nanoparticles. I am Manika Bhondeley. I'm a scientist in process and analytical development at Kudo Biotechnology, and I'm going to be the moderator for the webinar. We will be recording this webinar. So now I would like to introduce our special speaker today, the CEO and founder of Kudo Bio, Molly Bekogelin, and I would request her to introduce Kudo Biotechnology.
Hi, everyone. As Monica had indicated, I am Molly McLaughlin. I am the CEO and founder of Kudo Biotechnology. Just as an introduction to Kudo, we are a global mRNA CDMO. We are headquartered here in Needham, Massachusetts, outside of Boston. We started the company, I guess it's about 18 months now. The focus of the company is in personalized medicines for mRNA. We are a full service CDMO, meaning that we can do plasmid DNA. We also have an enzymatic cell-free DNA template. We have mRNA IVT, and then we can do LNP encapsulation, lyophilization, and fill finish. The key about this is that we can do it all in one location. So you don't have to worry about transferring your product from here to there, increasing risk, time, and money. So we are able to do this, like I said, all in one location. We have a facility right now in Shanghai, China, and we are building a facility here in the Boston area, which will be online in early 2025. We are the only CDMO team that has been able to do this all in one location with product that's in human clinical trials today. We are also an innovative company. We have 5 patents pending. We have adopted the most innovative and advanced technologies in our process. We are able to do UTR and codon optimization to increase protein expression. We have the lyophilization, which you'll learn more about today, which is all new innovative capabilities that we've done here at Kudo. We also are compliant with global compliance for not only ISO 9001, but all of your regulatory requirements for things such as FDA, the EU, MHRA, et cetera. So we are ensuring that we have global compliance, and we're constantly looking to see if the regulatory bodies have any new guidance or have any new comments that they want the industry to follow. And so we're ensuring that we are compliant. We also have track records of international tech transfers. So we have multiple projects where we have tech transferred from our clients' process into our facilities where we've done a scale-up optimization and then production of their process. We've also worked with several different technology platforms for things such as your T7, your capping analogs, and also LNPs. So we have a lot of experience with using customized lipid nanoparticles, lipids for doing the lipid nanoparticle encapsulations. So it really is looking at the most leading edge, innovative capabilities in manufacturing for these products. We also have a comprehensive QA and QC capability. We use a Zen QMS quality management system that governs everything we do from employee training all the way through batch record release and supporting your CMC applications. So this is Kudo in a nutshell. And with that, I'll give it back to Manika. I hope you enjoyed this webinar. I think that it's tremendously good information, and I hope you enjoy. Thank you.
Thank you so much, Molly. So before we begin, I would request everyone to submit the questions using the Q&A feature, and we will address it in the end. Now, without any further ado, I would like to introduce our speaker of today, Sara Townsend Miave. She is a principal scientist at Kudo Biotechnology. She has a Master's of Science in Analytical Chemistry, and she has years of experience with oligomers, small molecules, and mRNA-LNP drug development. And she has worked in multiple companies, specifically at Sarepta Therapeutics and Ayurvedic Medicine. Over to you, Sara.
Thank you, Manika, for your kind introduction, and welcome everyone to today's webinar on the lyophilization of LNPs. In this session, we will cover excuse me, sorry, somehow I got muted. Let me start over. Thank you, Manika, for your kind introduction, and welcome everyone to today's webinar on the lyophilization of LNPs. In this session, we will cover mRNA-LNP production, explain the lyophilization process, and cover some of the emerging trends for freeze-drying LNPs. Before we dive in, we will briefly touch on the overall production process of mRNA-LNPs. The process begins with a DNA design that will ultimately transcribe into the desired mRNA. There are two paths to this. There's the traditional path of pDNA production in E. coli, or a new shorter method designed by LinearRx, which produces the DNA by a PCR template. Kudo has the capability to do both methods. mRNA is produced from linear DNA by an in vitro transcription reaction. After purification, the mRNA drug substance is ready to be encapsulated in an LNP. A typical LNP uses 4 lipids: a cationic lipid, a pegylated lipid, a sterile lipid, such as cholesterol, and a phospholipid, such as DSPC. For the production process, the lipids will be dissolved in ethanol, and the mRNA will be diluted in an acidic buffer. These two solutions are then separately loaded onto a syringe pump microfluidic system or an HPLC T-mix device. The two liquid streams will converge in a narrow channel, and the rapid mixing will cause the lipids in the mRNA to self-assemble into this beautiful particle here. After this step, the ethanol and acidic buffer will be buffer exchanged into the final formulation buffer and cryoprotectant. The final product will then be fill finished. Depending on the scale, the labeling process may come after the freezing process, which adds complexity and therefore expense to this step because the LNPs cannot be allowed to thaw during labeling. mRNA LNPs are not chemically or physically stable at higher temperatures and are therefore currently stored at -80 in an ultra-cold freezer. Lyophilization has a strong potential to remove the need to freeze and maintain the mRNA LNPs in a frozen state until use. Centuries ago, the Incas of the Andes Mountains were believed to be the first to use a natural freeze-drying process. They would freeze potatoes and then let them dry in the low atmosphere of the mountains. The scientific method of freeze-drying was discovered in the early 1900s, adopted by the pharmaceutical industry in the 1930s for biological samples, and then widely used in World War II for food preservation. Personal anecdote: the first time I learned of the freeze-drying process, I was in a gift shop at NASA and begged my parents to buy me freeze-dried ice cream. Right here. Freeze-drying is not just a great way to extend the shelf life of materials. It can be advantage in transport because removing the water lightens the material considerably. This is useful if you are, say, an astronaut with a weight limit on your ship or a soldier in the army carrying your food on your back. Another advantage of freeze-drying is that it can remove the need for ultra-cold chain shipping. The cold chain is a logistics process for the transport and storage of pharmaceuticals. Additionally, each step in this process must be documented in the chain of custody paperwork. These documents are records of the transport and storage of the material and include states, who shipped it, who received it, how it was stored, the temperatures at the transfer time, and if there were any temperature excursions. For GMP pharmaceuticals, both the boxes and the freezers must have temperature monitors as well. There are many steps in this cold chain between the fill-finish step in manufacturing and the end use of the product. And a temperature failure at any one of these steps could be a costly mistake because the material will become unusable. Note that currently, lyophilized mRNA LNPs still need to be stored at refrigerated temperatures, but this is less costly and more attainable for developing countries than -80-degree storage. We've talked a little bit about the why of lyophilization, and now we're going to cover the how of lyophilization. As we all know, at normal atmospheric conditions, water exists as a liquid at room temperature and a solid below zero C and becomes a gas above 100 C. If we lower the pressure considerably, we can skip the melt step and proceed directly from a solid to a liquid in a process called sublimation, i.e., we can freeze and then immediately dry by removing the free water as a vapor. This is called the primary drying step in lyophilization. Some additional terms that are specific to lyophilization include cake, this here. This is the final solid that is obtained after removing the liquid from the material. This material may be amorphous or crystalline. However, most drug products are amorphous and will freeze into a glass instead of a crystal form. The temperature that this happens is called the glass transition temperature. And this temperature is influenced not only by the drug product, but the buffers and other excipients present. Knowing the critical temperatures, such as the glass transition temperature and the collapse temperature, is essential in order to ensure a successful lyophilization of the drug product. There are 5 main steps in the freeze drying process. After the LNPs are formulated into their buffer plus their cryoprotectants, they are filled into vials. The vials are partially stoppered and then loaded onto shelves in the freeze dryer. A partial stopper ensures that the water vapors can escape out of the top of the vial instead of being trapped inside to rehydrate the material. The material is then frozen to a temperature that is well below TG temperature, and the free water will become ice crystals. This temperature ramp is controlled because slow freezing will cause large ice crystals that will sublimate faster, and fast freezing will give small crystals. The large ice crystals are generally preferred because not only will they shorten the primary drying step, they also won't raise the temperature of the product and cause meltback. However, for some drug products, large ice crystals can be damaging to the product. So this is essential to understand and learn about your particular product and which would be preferred. The next step after the freezing step is primary drying. The pressure will be decreased considerably and the temperature increased, but still kept below TG/TC. And at this point, based on that temperature and pressure differential shown on the previous slide, the ice crystals will sublimate and leave behind some empty pockets. After this step is completed and all of this free water has been sublimated, the next step is secondary drying. For secondary drying, the low pressure is still maintained, and you may even lower it a little bit more, and the temperature is slowly ramped up to room temperature or above to avoid melting. Note that increasing the temperature to well above room temperature will shorten the drying time but could cause degradation of the drug product. This step tends to have a very long hold time, and what happens during this step is the desorption of bound water. After you've removed as much water as you can or you wish to, depending on your need for your product, the next step will be the finish step. In this final step, the chamber is returned to ambient pressure and temperature, and the vials are fully plugged with the stoppers. And the result is should be hopefully a pretty dry cake. A freeze dryer is needed for the process described on the previous slide. There are 2 main types of freeze dryers, including shelf freeze dryers and manifold freeze dryers. This freeze dryer in the center is a commercial scale up of an R&D freeze dryer shown here. Manifolds are used for bulk freezing, and the material must be frozen in this glass jar here before being attached and the vacuum opens. For shelf freeze dryers, the vials or tray of material is loaded directly onto these heat transfer shelves here. The freezing and heating of the vials is actually controlled by the shelves, not the chamber. The shelves are hollow and contain a thermal liquid which will cool or heat the material as needed. Shelf freeze dryers are typically equipped with two different pressure gauges. A manometer measures the absolute pressure, and a Pirani gauge will use the thermal conductivity of gas to determine pressure. The Pirani gauge will read higher if water is present due to the high thermal conductivity of water vapor during the sublimation steps. Therefore, the primary drying step is considered to be complete once the two different gauges are in agreement with each other, once they read the same chamber pressure. Lyophilization process development can be very complicated due to the many parameters that may need optimization. Parameters such as the design of the LNPs, which lipids, the lipid ratios, the N/P ratios, and which excipients to use. Excipients can include protectants, surfactants, and bulking agents. Cryoprotectants are typically sugars such as sucrose and maltose and protect the product during freezing and drying. The current hypothesis is that they help protect against particle aggregation by filling the space around the LNPs, and they may stabilize the structure. And they may stabilize the structure by taking over the hydrogen bonds from the water molecules as the material dries. Surfactants, such as polysorbent 80 and 20, coat the surface and can prevent aggregation or protein unfolding. They can also prevent the drug product from interacting with the container surface. Bulking agents help with the structure and appearance of the final cake, especially for low concentration formulations. Buffers maintain pH during freezing and after reconstitution, and may have an effect on the morphology of the LNPs before and after lyophilization. The fill volume is important because cakes that are too thick will have more trouble during the drying phase. Sublimation of ice crystals will happen more slowly, and the temperature of the cake could increase past the critical temperature and then collapse or melt. All 6 types of variables can affect the final product quality, often referred to as the critical quality attributes. Lyophilized products have additional set of CQAs to the liquid formulations. They are residual water as measured by Carl Fisher, and cake appearance. An ideal cake should have a homogeneous color and texture and should be the same size and shape as the liquid level in the vial. This is often described as elegant. However, it is possible to obtain a very ugly cake that does not meet these ideal standards for appearance and yet is still stable and meets all other CQAs. Reconstitution time really describes the ease of reconstitution. The cake should dissolve quickly with only gentle mixing or vortexing and leave behind no visible solids. Drug products are always released based on a full set of CQAs and the associated specifications. However, during process development, some assays may be tested to gain additional information on the formulation. And in addition, the drug product should be tested before and after lyophilization and the results compared. For mRNA-LNPs, the top attributes tested during process development are particle size, PDI, encapsulation, and mRNA integrity. Polydispersity index, or PDI, is a measure of the range of particle sizes in the sample. The more uniform the sizes, the smaller the PDI. Typically, larger particle size are thought to be less stable and can be more prone to mRNA leakage. mRNA leakage can be seen by the lowered mRNA encapsulation efficiencies. For LNPs, the most important additional tests for freeze-drying process development are zeta potential and transvection efficiency. Zeta potential is a measurement of the electric charge of the surface of the particle. Reordering the structure can have an impact on this zeta potential value. Next slide. Next, I'd like to highlight two recent publications in the field of LNPs. This 2023 paper by Moderator et al. Investigated the impact of cationic lipid ratio to mRNA ratio on the CQAs of mRNA-LNPs before and after lyophilization. For their study, they used the C12-200 LNP and investigated tris, PBS, and phosphate with 12.5% sucrose. The first thing I would like to make note of is their term of weight ratios of lipid to mRNA. Instead of using weight ratio, we can think of this as the more traditional N to P ratio, moles of nitrogen in the lipid versus the moles of phosphorus in the mRNA. The weight ratios they are using calculate out to 3.1 and 6.1, which are typical weight ratios that are normally used for LNPs. This page displays the particle sizes for their three formulations at the two ratios. Note that for all formulations, the particle size in PDI did not significantly increase and even decrease for some of their formulations after lyo. This is atypical and could be due to their particular lipid they used or their spin drying process. The next page is a continuation of their CQAs for their investigation. Here is their encapsulation efficiency and zeta potential data, again for the three formulations of the two weight ratios. For the study, Moderator et al. The percent encapsulation and transfection efficiency significantly for PBS versus the phosphate and tris. The PBS buffer had the largest decrease in zeta potential as the others. The 20-to-1 lipid to ratio performed better than the 10-to-1 in terms of percent encapsulation and zeta potential. So here is the zeta potential, as you can see, very negative for this is their PBS. And it's similar over here for 10-to-1. And their phosphate versus tris isn't performing that poorly, but the PBS only performs much more poorly than the others. So, in to the typical CQAs, they also cryo-EM observe the morphology of the nanoparticles before and after a lyo, two of their ones. And in fresh PBS buffer, they observe these structures here, which are called blebs. And the nanoparticles change to multilaminar structure in addition to some blebs. The tris, on the other hand, initially called amorphous cores. After drying, those are present, and now blebs could also be observed. For their 4C stability, Moderator used the 10 millimolar phosphate millimolar tris at the 20-to-1 weight ratio. Only they also use the tris formulation for their study at room temperature and 37 degrees. At 4C, the lyophilized mRNA-LNPs and tris maintained both their encapsulation and efficiencies for the 12 weeks, but increased after only 1 week. The tris formulation at room temperature behaved very well and did not show significant effect in transfection or encapsulation efficiency, particle size. So this is transfection efficiency, encapsulation efficiency, and size. Tris is behaving even steady. Again, not even seeing large growth. Everything maintaining the same. Phosphate actually looks okay. Tris, yeah, so the big in the tris here is 7 degree. This study in it by 12 weeks particle size growth. But the promising thing is that their data looks really good for 12 weeks. Okay. One question often comes up regarding lyophilization of LNPs is the effect of lyophilization on in vivo performance. Moderator were able to demonstrate the performance of the lyophilized drug product favorably to the LNP. So this figure, the in vitro transfection efficiency, looks very good. Figures D are on the in vitro transfection efficiency. You can see this one here is fresh, this one here is lyo. They look very much the same. Even here in the RO, the total flow, they're very similar. So this is very promising. For that they bring 20 millimole less formulation. So 2024 paper by Alejandro et al. Also investigated the use of tris and PBS buffers for their formulation. Their cationic lipid was SM102. Similar to Mulewater et al., they observed that the transfection and percent encapsulation were lower for the PBS buffers and the data potential more negative than tris. Mulewater et al. Grew considerably after lyophilization, with PBS having the most substantial. So as you can see, their pre-lyo is, I don't know, 80 nanometers or so, and all of their post-lyo here is considerably better, with their PBS the worst in the vertical growth. And you can also see here, similar to Mulewater, the transfection efficiency is positive here. It looks okay, but definitely, as you can see, it's pretty much, there's no change, whereas there is a little drop in PBS. After their initial screening optimization of their freeze-drying parameters, they chose 5 millimolar and 20% multi for their stability. Despite the large particle size at =0 for their lyophilized LNPs, samples were able to maintain their size, PDO, transcription efficiency, percent encapsulation for 12 weeks. They dropped below 1%, but still acceptable. The green line here is their lyophilized data. PDI for decrease here, but so particle size. This is their particle size. It doesn't change much. It's high, starts through it, but then it stays throughout. Their integrity does drop some by their 60. I mean, 60 weeks is more than a year. 60 weeks, but well, probably about 6%, which is Zero et al. Also published on Crero TEMD. If we look carefully at the pictures, we can see a correlation between these results for Mulewater. The non-lyophilized appear to be solid. Here's the non-lyophilized. This is, again, they're using tris. They're using tris. They have a completely different lipid. Their cores very similar to Mulewater. After lyophilization, there are some blood structures that come in, and over time, it seems like there's more and more of these bleb structures. Definitely, and there's some aggregation here for the 7. One. Kudo Biotechnology has also experimented with the lyophilization of LNPs. For our formulations, we use the and we tried 4 and 3 and heaps buffer with 5% and 12% sucrose. We had a good success conditions and chose 4 millimolar with 12% sucrose for a scale up. As you can see from the data, we were able to scale up from an R&D freeze dryer to a pilot scale freeze dryer with substantial loss in encapsulation or integrity and only minimal growth. We also tested post formulation against the liquid sample, using the liquid sample reference, and 88%, which is a very acceptable value. Success of our mRNA-LNP utilization, Kudo took it one step to lyophilize circular mRNA-LNPs. Again, all 3 conditions worked very well, and there was only minimal size growth. Note that in addition to Kudo investigated heaps buffer in addition to TRIS, had not been explored by others. Kudo also tried each formulation at 3 different concentrations and did not see a significant difference, which is very useful data. Summary findings. Review of several journals lyophilization of mRNA-LNPs revealed some interesting things. 1, it is possible to lyophilize particles without obscene particle size growth. 2, lyophilization is a strong pathway to removing the need for our ultra-cold chain. 3, salt-based buffers should be avoided. TRIS at any concentration is a strong contender for a buffer for LNPs, but more buffers should be investigated. TEM and zeta potential about the structure of the LNPs in various situations and before and after lyophilization. The final key to understanding thermal LNPs lies within the internal structure of the LNPs. Okay. Thank you, everybody, for your attention today. This concludes our lyophilization LNPs talk part of the.
That was a great talk. I think now we can start the Q&A session. And the first question we have is, okay, so are the parameters optimized for small-scale lyophilization directly transferable to larger batches? What might arise during scale-up and how can they be mitigated?
This is a really good question. The R&D scale, the scale equipment, known to have very different heat transfer activities, so that they're not going to behave similarly to try to not just it's not an easy just add 10 minutes. It's nothing like that. So but there are mathematical models that a person, if you understand the transfer of the one instrument versus the other instrument, if you understand all those parameters, there are mathematical models where you can put those parameters in and help you model what you should be for your work and get very close.
Thank you, Sarah. Okay, so I think time for 2 more questions. The second question we have is, means that some formulation changes zeta potential significantly. How does it correlate with product quality?
being investigated, but it is something I'd like to see more. These recent papers, the first patient where potential is really explored, and I do think that the hypothesis is that if your changes very between before and after lyo, that you may have a more stable particle, but that is just a hypothesis and we need more data.
All right, thank you, Sarah. Okay, so the next question here is, how does the particle size distribution and encapsulation efficiency change upon reconstitution?
So upon reconstitution, your particle size and in your distribution can change from before and after lyo considerably. Some of the data I showed earlier, their particle size was about 80 nanometers. And for the various formulations that they explored, it went up to 200 or 300 nanometers after.
Okay, yeah, thank you. All right, so let me see. Okay, so the next question is, what solvents or buffers are most effective for reconstituting the lyophilized product? Do they restore the original particle size PDI and encapsulation efficiency?
This is a great question. Typically, in most of the research papers, they used Wi-Fi for reconstitution. However, based on some previous experience that I have from RVAC, it is possible that reconstituting with a salt-based buffer versus Wi-Fi could actually give you a smaller particle size upon reconstitution. However, this is again one of the things that I think needs more data and more exploration to really understand.
Right, yeah, that makes sense. So do you think that smaller particle size, that is a good thing?
Actually, so there's another paper I read, it's here in the references by Fan et al. And they actually correlate particle size and encapsulation and to transfection efficiency and integrity to transfection efficiency. And they actually found there's a stronger correlation between your integrity and your encapsulation than there is between particle size and transfection efficiency. So it's long been believed that you need to have smaller particles in order to get to where you need to go, in order to get in the cell and drop your payload. But there's data out there that kind of contradicts that.
Right. Thank you, Sarah. Okay, so I think we can take maybe 2 more questions. Let's see. So the next question here is, how much time does lyophilization add to the drug product manufacturing and release?
So for manufacturing and release, I would say, so lyophilization can be a very long process. The drying, it can take 7 days or more to dry, from freezing to drying for products. But the actual release part of this is not going to be any longer than your typical release specification time length. So I would say maybe 2 weeks longer if you wanted to add lyophilization.
Okay, yeah, that's not bad. Okay, thank you. Okay, so the next question is about residual moisture content and residual and reconstitution time. Like, how do we test that?
That is more an observation
Okay.
than a physical attribute that's tested.
Okay. All right. Okay, so next question is, how do the freezing rate and drying temperatures influence the physical characteristics of the LNP?
If only we knew. This is something that, again, this is something that the field is currently researching and trying to figure out. One of the papers I did cover, they they narrowed down their formulation, and then they went back and they tweaked some of their drying parameters, and they were able to get better data after doing that for their LNP than before they did that. So there is definitely an effect. What that effect is, if it's an effect for certain lipids and not others, if it matters more if you're using tris or keeps or this cryoprotectant or that cryoprotectant, there's so many different parameters that it's hard to pinpoint exactly which thing each step is going to affect.
Okay. Thank you, Sarah. All right. So the next question here is, so if we have a very dense solution that keeps melting after lyo, what step can be taken?
If you have a very dense solution that keeps doing what after lyo?
That keeps melting. So it keeps melting after lyo. What steps can be taken?
Okay, if you have something that is melting after lyo, that's interesting. Okay, so what that means is that your glass transition temperature is now the same as your room temperature. And you might want to try different cryoprotectants, because cryoprotectants actually change the glass transition temperature of your product. So some sugars have like a higher glass transition temperature. So investigating different sugars, or even sometimes they mix different sugars in order to help with this effect.
Do you think that's better, like mixing different sugars and then using that as a cryoprotectant, or just using like sucrose?
I think it all just depends upon your drug product.
Right. Okay. I think it's about time. I don't think we can take more questions, but yeah, I do see some questions there about lyophilization capabilities and timeline. I think about that, our BD team can get back to you. So before I close, I do have a few announcements. So we will publish the white paper of today's webinar on our website, and we will also send the link to everyone registered today. And for this work, we also have a patent, which is pending submission.
Oh, it's submitted.
So it's submitted. Sorry about that. So with that, thank you so much, everyone, and have a great rest of your day.